now. Okay, guys, welcome to our first biology revision session. We're going to be doing topics 2.1 to 2.5. And yeah, let's just get right into it. So, just a little information about my sessions. They're going to be recorded. I'm recording right now, and they're going to be posted to a YouTube channel that I made specifically for revisions. Um, I'm not going to be looking at Discord for the remainder of this session because I can't, because I'm looking at my speaker notes. And I'm not going to be taking any questions during recording just to make sure the recording is all good. The link is in the PDF for anyone who's using these to annotate as I go, so don't even worry. And just for a quick overview, I'm going to be going over topic 2.1 to 2.5 today, which is both standard level and higher level. And then for the next 3 sessions, I'll be doing 2.6 to 2.7 in conjunction with topic 7, and then the rest of them are just kind of on here. So yeah. All right, so getting right into it, we're going to start off with topic 2.1. Here are your learning outcomes. You can read these on the PDF or just in the video later to help you kind of guide yourself along your study process for this topic, but I will be doing the rest right now. Okay, so just a quick general overview. Molecular biology is a field of study exploring molecular interactions in biological systems. So basically all of the chemistry that sort of goes down in your body, an organism, literally anything in biology. And the kind of main things that we look at are structure and function. So structure can be something like cellulose, which is a special biomolecule in plants to help them stay structurally integral. And function, so something like an enzyme, what an enzyme does, what its role is, and also crosstalk. So how all of these things interact with one another in biological systems. So this can be something like signaling, whether it be extracellular or intracellular, all that fun jazz. Now, to briefly touch on regulation, biological processes are tightly regulated by enzymes. And we're going to get into enzymes in 2.5, but this is just kind of like an overview. Expression of enzymes are regulated through gene activation and inactivation, so you guys should know this through what you guys learned in terms of the central dogma, and I'll just quickly draw that out. We have our little DNA molecule right here, and we're going to go through transcription and then translation, and then we're going to get, we're going to draw a little enzyme here. That does a particular function, that does its job. And there can be different signaling molecules. Let's just draw a little circle, and this will be our signal. This signal can be intracellular and extracellular, it could be either. And the way it can affect this whole process is it could affect the process of transcription. It could activate it, it could inactivate it. So this could be positive or negative. Or it can affect the enzyme itself. So it can affect its function, either positively or negatively. So yeah. Now that we're kind of done that, we're going to talk about organic compounds because there's a lot of chemistry that goes down in molecular biology. So understanding the basics of it is the most kind of important thing. So what is an organic compound? Well, it's something that contains carbon and is found in living things. Now, there's obviously going to be exceptions to this, and they're right here. So carbides, well, what's an example? An example would be calcium carbide. This is found in living things, but it's not an organic compound because they're not derived from... It's kind of arbitrary, but these are the only 3 that you need to know. So carbides, and this is the example, carbon oxides, so you know one already, carbon dioxide, also carbonate, so carbonic acid, and cyanides, so you know this as CN-. And then there can be other chemicals that have this as a functional group, etc. So these are your kind of exceptions, and this is just the basis of what organic compounds are. we're going to look at a theory about organic compounds, and it's called vitalism. This is a pretty old theory, and it's the idea that living things have a vital force needed to make organic molecules. So basically, like they can only living things can make organic compounds. But this was disproved by Friedrich Wöhler, I think that's how you say it. And in a lab, he reacted cyanide with ammonia, and he made urea, which was previously thought to only be able to be produced by us, but he made it in a lab. So conclusion, we don't need living things for organic compound synthesis. We can make them in a lab. So that's kind of the main thing I wanted to mention there. Okay, so to kind of show the foundation of all this, we're going to talk about carbon. So carbon forms the basis of organic life, and there's a couple reasons for this. The first reason is it can form 4 covalent bonds. So I'm just going to draw a little carbon atom here. And it has 4 little valence electrons and 4 missing. So that means it can bond to 4 different things. An example, a carbon to carbon bond. So they share their electrons, their covalent bonds. And another example could be carbon to oxygen. Again, they share their electrons. And another example is carbon can have double bonds sometimes. So it can have something like this. And this is still 4. 1, 2, 3, 4. So you count a double bond as 2. And when it's a triple bond, you count it as 3. So that's one reason it can form 4 different bonds. And the second reason is that carbon to carbon bonds are very stable. So if we look at this over here, this is super duper stable. And that's why we can have super complex molecules like fatty acids. So as you can see, there's a lot of carbon to carbon bonds here. And this is like present in pretty much every biological system. So these are the 2 reasons why carbon is so important in organic life. Now, with that being said, we're going to go over the types of macromolecules. So another way to say this is biomolecules. And I just mean the major groups of molecules that are found in living systems. So first is carbohydrates, and they are the most abundant biomolecule in nature. And their molecular formula is CH2O, and there's an N. So if you have something like glucose, it's C6H12O6. So N would be 2 in this case. And yeah, they act as a source of energy storage. So this could be something like glycogen in the liver. Recognition, that could be something like glycoproteins. How if a cell touches like the glycoproteins of another cell, it can recognize it. What am I writing here? And structure. So structure could be something like cellulose, like I mentioned before. So carbohydrates really have a lot of different kind of functions. Next, we have lipids. So lipids are nonpolar and hydrophobic. That's their kind of characteristic type beat. Their main sort of bond is CH, so carbon-hydrogen bonds, and also carbon-carbon bonds. Those are the main sort of bonds that we see in lipids, and there are many, many, many types. So this makes them not necessarily a polymer because they're just so diverse. There can be something like fatty acids, cholesterol, so just sterols in general, and also something like triglycerides. These are all different kinds of lipids. And similar to carbohydrates, they act as a source of energy as well as energy storage. So the storage would be in your adipose tissue, so the storage of triglycerides in your fat tissue. And then as a source of energy, they're more of a long-term source. So I would call this short-term energy storage. and I would call this one long-term energy storage. Another function they have is cell membranes. So I'm sure you can imagine the different types. So phospholipids is one of them. It has lipids in the name. And another one is cholesterol, present in mammals. And lastly, signaling. There's a lot of different types of lipids involved in both intracellular and extracellular signaling. So, yep. Next, we're going to do types of macromolecules. Oh God, I already said that. The titles are messing me up. The last two are proteins, and proteins make up over 50% of cellular dry weight. So you have a lot of proteins. And the types of atoms they're made out of is C, H, O, S, or N. And you can remember this as chosen. Proteins are chosen by your body to do different functions, and they have all these types of atoms. So an example of their functions is catalysis, so enzymes, and also structure. So you have stuff like collagen. Collagen helps support a lot of your tissues in your body, like your lungs. So yeah, then we have nucleic acids finally, and these are made of the atoms C, H, O, P, N. And you can remember this as choppin'. So I guess whenever you need to make, I don't know, how can I think of an analogy for this? I don't really know if you can relate a knife to DNA, but choppin' is something you can use to refer to nucleic acids. I don't know, I guess exonucleases are chopping the nucleic acids. I don't know. But yeah, their function is mainly the storage of genetic information. So this is going to be through your DNA. And, well, okay, RNA is not really for storage, but we're going to include that here. Well, we can just put that here. DNA and RNA, those are the main nucleic acids, is what I was going to say. And then the central dogma, which anytime I say central dogma, I mean this. So DNA is transcribed to RNA, which is translated into protein. So this is what I mean every time I say central dogma. It's just referring to this process of transcription and translation. So this is something that nucleic acids are incredibly important in. They make up like 2/3 of the whole process. So yeah. Next, we're going to get into how these kind of organize. So large macromolecules or biomolecules are made of polymers. So polymers have a subunit, and all four of these types of macromolecules can act as a subunit in some way. Lipids are kind of an exception. So carbohydrates are monosaccharides. So an example here is, well, first I'll just break down the word. Mono means one, and saccharide means sugar unit. So one sugar unit, I'll draw this a little better here. And an example of this is glucose. So I'll just draw this. So one of the things you're going to have to know for the exam is how to draw glucose. So yeah, that's an example. And they act as building blocks, of course, they're subunits to disaccharides like lactose and polysaccharides. So this is something like cellulose or glycogen, like we talked about before. And these are both very important in their function. So usually they're present in ring structures, and this is what I kind of talked about here. So this is kind of a ring structure. Sometimes it's linear, but in solution it's always cyclic, or like a ring structure form. And there can be different arrangements of these OH groups. So if you look at these right here, some of them can be flipped. So an example of this is alpha and beta glucose. So alpha glucose would have the OH here. And I'll just draw the rest of this. And then something like beta glucose would have the OH here. This will all make sense kind of as I break it down even more, but these two you're going to have to know how to draw on the exam. Let's see. Okay. Oh, and also ribose. You're going to want to know how to draw ribose. Let's see if I can fit it on here. This is ribose. So they can have different amounts of carbons is something I was going to say. And ribose is another one of the ones that you're going to have to know how to draw. Okay, moving on. Lipids. So lipids are not a monomer. They do not polymerize, not really like a thing for them, but they can arrange in different kind of ways. And an example is fatty acids. So we're just going to draw one right here, and they're basically very long hydrocarbon chains, and it has an acid group. So this would be a saturated fatty acid. The abbreviation I'm going to use is FA. This you're going to have to know how to draw. For your exam. And you don't have to know the name, you don't have to know anything else, just besides the fact that it's a hydrocarbon chain with an acid group at the end. That's all you're going to have to know in terms of how to draw. And there are other types of lipids too, and just to illustrate the complexity of how some of them can be, here's cholesterol. You do not have to know how to draw this. But I just showed it just to show you that there's different kinds of lipids and they can look very widely different from one another. But yeah, you should be able to recognize these though. Just know that sterols have a lot of different ring groups, I guess. Okay, the third one is proteins. So the monomer for proteins is amino acids. So one thing you're going to have to know is how to draw a generic amino acid group. And the first thing they have is a central carbon. So this is our little central carbon. And then they have a carboxylate group, or carboxyl. And that's going to look like this, COOH. And another thing they're going to have is an amino group. And this is going to be NH2. And then there's going to be a variable group, an R group. Anytime you have to draw one of these on an exam, you're going to use R as the variable. And let's see, I think lastly, we have an H. So the H is there to fill out the rest of the bonds carbon needs, since carbon can have 4 bonds. So this is an amino acid. And the R group over here determines the properties of the whole amino acid. And the abbreviation I'm going to use for amino acids is AA. So this whole thing, the whole properties of it, are determined by the R group. And when you have different amino acid chains, different combinations of R groups will make different types of functioning proteins. So yeah, that's the subunit. Lastly, there's nucleic acids, and their subunit is nucleotides. So this one I don't believe you have to know how to draw, but you will have to know kind of the basic structure so you can recognize it. So first we have a pentose sugar, and this one actually just so happens to be ribose. I'll just draw this out. Okay, so that's the basic structure. This would be a ribonucleotide. One second. Actually, yeah, this is the right one. So when it has an OH, this would be part of RNA. And when it has an H, we're just going to number this. 2 prime, 3 prime, 4 prime, 5 prime. So I'm sure you've seen the numbering on these. The one starts where the base is connected, and it goes 2 prime, 3 prime, 4 prime, and then 5 prime is where the phosphate is connected. And then this here is a nitrogen base. And this one would be 1 prime. And then this 3 prime OH is what's used to bond multiple nucleotides together. And this one will attach to a different 3 prime OH. So yeah. Anyways, as I was saying, this 2 prime OH group can either be an OH or an H, and that will change whether it's an RNA or DNA. If it's an OH, then it's an RNA, and if it's just an H, then this would be DNA. The reason why is because the D in DNA is deoxyribo, and deoxy means no oxygen, and that's referring to the fact that there's no oxygen in the 2 prime carbon. It's just an H. So that's why the naming from there is what it is. And these nitrogen bases they can be different in DNA and RNA. An example is uracil versus thymine, or U versus T. U is only found in RNA, and T is only found in DNA. Don't worry about the if this is kind of all over the place. This will be very kind of prominent in topics 2.7 and 6, I think, which is the genetics part. But yeah, that's the subunit for nucleic acids. Next, we're going to get into polymers. So more of the specifics of how these subunits arrange and become how they are in biomolecules. So starting with carbohydrates, again, these are composed of glycosidic bonds. And this means a bond between sugar units, kind of like this. So sugars, I forgot to, I'll cover this later, but the numbering for sugars are, oh God. The numbering for sugars goes 1, 2, 3, 4, 5, and 6. So it always starts at the right of the oxygen. And if we follow the same pattern here, this is an example of a 1,4 glycosidic bond. And we'll get more into this later, but that's kind of what it looks like. And you can have things like, I'll just draw this for simplicity. You can have stuff like disaccharides, and an example of this is lactose. And you can have polysaccharides. So an example of this is amylopectin and cellulose. And these can be branched or unbranched. And whether it's branched kind of determines how it's used. So branched includes stuff like cellulose. So I'd rather just kind of, cellulose and amylose. And then unbranched, oh gosh. Sorry, I meant unbranched for cellulose. Just woke up, sorry guys. Unbranched would be cellulose and amylose. And then branched would be something like amylopectin and glycogen. So glycogen and amylopectin are used as sugar storage, and so is amylose, and cellulose is used as structure. So all of these kind of have different functions. And the properties depend on the monosaccharide subunits. Something like lactose is made of glucose and galactose, which are both monosaccharides. And if it was something like glucose and glucose, it would make maltose. So different combinations make different functioning carbohydrates. Okay, next is lipids. So there's 3 main types. So simple would be something like a triglyceride. So this is when 3 fatty acids combine together and attach to a glycerol to make a triglyceride. Tri means there's 3 fatty acids, and glyceride means it's all attached to a glycerol. So this is kind of the main simple lipid. So simple lipids is the biggest group and is triglycerides. Next, is compound lipids. So this is going to be something like phospholipids. And phospholipids are kind of similar to a triglyceride, except the difference is there's only 2 of them. And this is still glycerol, by the way. The bond can just go the other way. They can rotate around. And this is actually not going to be a fatty acid, but it's going to be a phosphate. And then this can be attached to a variable group. So this would be 2 fatty acids and a phosphate. So another name for these is glycerophospholipids. So compound, they have a phosphate. And then derived, these would be sterols. For example, cholesterol. And also steroids. Sterols are part of the steroids class. So something like cortisol or testosterone. All of these come from cortisol. They're derived. So things like sterols and steroids are derived lipids. So those are the 3 main sort of classes. Next, we have proteins. Now, the polymer of proteins is polypeptides. So this is when all of the amino acids come together to form multiple bonds, and and all the bonds between amino acids are called peptide bonds. So I'll just show you kind of what these look like. I'll draw the NH2 or amino group here, the central carbon, and the carboxylic acid group. I'm going to be a little more specific with this one. This is what COOH means. And then we have our R group. Then we have our H group. And if we combine with another amino acid, so R1 and R2, just to show that they're different, if these combine, well, one of the OH here and an H here is going to leave as water. And the result will be, oh, this is going to be pretty tight. The result is going to be a dipeptide. And these leave as water. So the OH and H here combine to make a water. And together, this is called a condensation. And whenever you hear condensation, it's always going to mean you're going to get a water out of it. So whenever you have condensation, you're going to make water. And this is how a dipeptide is formed. And you're going to have to know how to draw a peptide reaction. And I'll get into this later. And on the bottom here, amino acid chains are folded and then become functional proteins. So it starts out as something like this, and then it's folded into a complex shape that will be a functional protein. So out of this slide, just know that the polymers for proteins is polypeptides, which is made up of amino acids. And the bonds between them are peptide, and that's how they form a functional protein. Lastly, we have nucleic acids, so DNA and RNA, like I said before, and nucleotides form phosphodiester bonds. And a lot of these together make different nucleic acids. So I'm just going to draw an example here, and I'm going to make this super simplified. Might want to draw this closer in the middle here. These bonds here are called phosphodiester bonds. When the phosphate on the 5' group of one is linked to the 3' OH of another nucleotide, that's going to be a phosphodiester. There's a phosphate, and then there's 2 ester bonds, and that's going to be a diester. And together, when you have all of these bonds in tandem, they're going to form helical arrangements. So RNA can look something like this, and DNA obviously has the double helix. And this is because of how the bases interact with one another. Okay. Next, we're going to go to metabolism. So, what is metabolism? Metabolism is all the chemical processes that occur within a living organism required to sustain life. So, all the chemical reactions that you need to live, that's your metabolism. So, you may have, this word may be familiar to you, but something like glycolysis is a pathway that's in metabolism. And metabolism serves two key functions. The first one is to provide a source of energy for cellular processes, and this is called catabolism. We're going to get into these words in the next slide. But an example for this, like a cellular process, I'm going to put the arrow here. Yeah. For cellular processes, this energy is really important, and an example of one is reproduction. And growth. You need a lot of energy for these processes. So catabolism, which is a type of metabolism, gives you the energy required for this. The second function is it allows intracellular synthesis of different materials, and this is called anabolism. And what kind of materials? Well, we actually just covered them, a lot of macromolecules. So nucleic acids, polysaccharides, even lipids, or even glucose. Any kind of synthesis from smaller molecules to bigger molecules is called anabolism. And this requires energy. Now we're going to get into these words that I mentioned. So the subdivisions of metabolism, there are two. There's catabolism, which is what I mentioned, breaking complex molecules into simpler products. And an example of catabolism is glycolysis. Taking glucose and breaking it down to get pyruvate. Pyruvate's a 3-carbon molecule, and glucose is 6 carbons. So clearly we've taken a complex molecule and broke it into a simpler one, and you get energy out of it. What is this energy? Well, ATP and NADH. I'm not going to get ahead of myself here, but the most important part here is that it produces energy when you break down a bigger thing into a smaller thing. Now, you're probably wondering what these words are, and exergonic means it produces energy, and oxidative means you lose electrons. So the molecule you're breaking down loses electrons. And the abbreviation for electrons I'm going to use is this E with a minus on top. But this is really important because these electrons can be used to make NADH, and that can power ATP synthase through the electron transport chain to give you energy. And one really common process in glycolysis is hydrolysis. If we have this polymer, for example, and we add water, this water is going to get in between here, for example, and break this bond. And it can also get in between here if you have two. And the result is going to be this. So these shapes are just to show monomers. So hydrolysis, whenever you hear this, you're going to use water. So hydro, water, lysis, breaking, water breaking. So this is a very common reaction you're going to see in catabolism. Our second subdivision is anabolism, and this is the second one, where you synthesize complex molecules from simpler precursors. And an example of this is nucleic acid synthesis. So when you're combining multiple nucleotides to make DNA or RNA, mRNA specifically during transcription. And whenever you have reactions like these, you're going to have something that's endergonic and reductive. And these are exact opposites of the terms we just talked about. So endergonic means it requires energy. And reductive means you need electrons. You need to put in electrons. And this could be something like NADH. So putting these electrons in will help you build these molecules. And these two words are what describe anabolism. Now, a very common reaction you see in anabolism is condensation. So let's go back to those shapes. We're just going to draw the triangle and the square this time. And if you combine these together, well, what's going to happen? This OH and this other H, or it could be the other way around, is going to leave as water. Now, we've seen this before when I talked about peptides. So whenever you see condensation, think making water. You're making water while also combining two monomers together, or multiple, to make a polymer. And that is topic 2.1. Good job, everyone. We got a few more to go. So this is topic 2.2, which is water, and here are your learning outcomes. Okay. So starting off, we're going to do the structure of water. Well, what does water look like? Water has an oxygen, which has two lone pairs, and then two pairs of electrons shared between oxygen and hydrogen, and you get H2O. Now, the reality of this kind of bond is that the electrons are not equally shared, and that's something that's called polarity. So, in reality, the electrons are going to be very close to this oxygen, but then the H's are going to be very kind of far away. And this is called polarity. The electrons are closer to the oxygen than the H, and this is because of electronegativity. Oxygen is electronegative, and this just means it loves electrons. So if it really loves electrons, it's going to want them really close to it. So we're going to have something like this. And this creates partial charges. And the way we refer to them is delta. So partial negative and partial positive. Partial positive. When you have negative, that means you have a lot of electrons. And oxygen has a lot of electrons near it because it's electronegative. So it's going to be delta negative. Hydrogen has like no electrons. Electrons are very far. And it makes it partially positive, so delta positive. So, just to summarize, because oxygen is electronegative, its bonds with hydrogens in water give it a partial negative charge or delta negative. And then hydrogen in the bonds with water have electrons very far away from the hydrogens, making it partial or delta positive. Another feature about water that's super duper important is hydrogen bonding. So this really strong polarity of water with electronegative oxygen makes it a really great candidate for hydrogen bonding. And this means in an environment with multiple water molecules, we're just going to write the partial charges. The partial positive of H and the delta negative of another oxygen in another molecule are going to interact very closely. And this is a type of intermolecular force. And this is called hydrogen bonding. And this one can interact with another oxygen in another water molecule, and so on. And this continues. Hydrogen bonding exists in molecules or atoms with oxygen, nitrogen, or even fluorine. So these ones you're going to have to know. Oxygen, nitrogen, fluorine, these are the ones that can have hydrogen bonding. We're just going to draw this process one more time, just to show you the complexity that it can have. And to show you that the lone pairs on oxygen are the things that are interacting. Oh, and something I wanted to mention, because oxygen has these little lone pairs, this takes on a bent conformation. But yeah, just gonna show a little more of this. And you know what? We're gonna spice it up a little, and we're gonna make these a different color because I feel like this might be a little confusing. Let's make it blue. So anytime you have a hydrogen attached to oxygen, nitrogen, or fluorine interacting with another one of those electronegative atoms in a different molecule, you're going to have hydrogen bonding. Another thing water can do is it doesn't have to hydrogen bond with only itself. Like I said, it can bond with other ones too. And an example of this is ammonia, just as an example. It can have hydrogen bonding. Or an amino acid. Anything kind of with nitrogen or fluorine or oxygen. And yeah, this is hydrogen bonding. Now we're going to talk about the different types of properties of water. And the first one is thermal properties. So one's to do with energy. To start off, it takes a lot of energy to vaporize water, which means turning it to a gas. And the reason why is because of hydrogen bonding. In something like water, these bonds are very strong. And in liquid water, in order to get this to be a gas, you need to break these bonds. And breaking them takes a very large amount of energy since they're very strong and stable. Destabilizing them takes lots of energy, which is why water has such a high boiling point. Now we're going to compare this to something like methane. Well, what does methane look like? It has the chemical formula CH4, and its structure looks like this. Now, CH The bonds between carbon and hydrogen are polar, but they're not polar enough to create a electronegative interaction strong enough to call it hydrogen bonding. So it's not overall, it has 4 bonds that are evenly distributed, which creates a net non-polarity, a net 0 in terms of its dipole moment. Water, because it's bent, these hydrogens and this delta positive creates a net dipole moment going downwards, and that makes it polar. Now, if we compare methane to water in terms of its thermal properties, starting with melting point, well, water's melting point, I'm sure we know this, 0 degrees Celsius. And methane is very low, around negative 180 degrees Celsius, somewhere around there. And it's very low because it doesn't take that much energy to separate their bonds, their intermolecular forces, because they're not hydrogen bonds. So there's no hydrogen bonding because it's non-polar. And because of this, it takes very little energy, if any, to even separate them. So that gives it a very low melting point. Now the boiling point of water, well, we know this, it's 100 degrees Celsius. I hope we know it. And methane, well, it's also going to be super low. I actually don't think I remember, but it's way lower than that. All you need to know is the relative things. So with methane, both its melting point and boiling point are significantly lower than water, and that's because it has no hydrogen bonds. And that is because it's non-polar. So this polarity gives water very important properties, and that allows it to be such an important molecule in life. In terms of thermal properties, something we're going to talk about is sweat. So water is a very good coolant. What does that mean? Well, we talked about how separating these water molecules in terms of their hydrogen bonds takes a lot of energy. And this can be utilized in something like sweat. When we exercise, this is going to be our skin. During exercise, you're going to produce heat. And when you have something like the air, well, the air doesn't really absorb that energy coming off of your skin. And this can cause overheating. And that's why your body has a very important mechanism called sweating. So we'll draw this again, and it's also during exercise. You're still going to give off that heat, but then you're going to have something like a water droplet, and all this heat is going to go here because it takes a lot of energy to separate the bonds between water molecules. So all that energy is going to go there. And water, therefore, absorbs energy or heat very well. And that makes water a very good coolant. It absorbs heat during exercise, and that's why we sweat, because water is such a, like, it absorbs energy very well. That's what I'm trying to say. And this is an example you're going to have to understand for the exam. Our next property is cohesive and adhesive properties. So this is very relevant in plant biology, as you know, topic 9. We love that one. So starting with cohesion, cohesion is when water has interactions with itself. And this is not special at all, just hydrogen bonding. And this is what keeps water together with itself. It likes to stick to itself. And this is called cohesion. Adhesion is when water sticks to something else. So not water. So adhesion means water likes to stick to other things. I'm just going to move this down a little bit. An example is surfaces. Because one thing cohesion and adhesion form or help to create, so I'm just going to, I guess I can, next slide. So one thing cohesion and adhesion create is something called capillary action. We're going to have this tube, and we're going to have a water droplet. And it's going to move up. So what causes it to climb? Well, in order for it to climb, these water molecules are going to stick to the surface. So this is called adhesion. And the type of intermolecular forces involved could be hydrogen bonds, could be polar interactions, or it could be London dispersion forces or van der Waals. I think mainly these two. These interactions help it stick to surfaces. So this could be something like a plant vessel or whatever. And this is what allows the water to climb. Well, what drags the rest of the water with it? Well, these water molecules are going to just follow these in the interactions. And this is called cohesion. And the types of interactions here are hydrogen bonds, which are a type of polar interaction. So capillary action. And this adhesion and cohesion in water, as another example, is why water droplets look like this on a surface. Because water is sticking to itself a lot, and this creates surface tension. Now, surface in this case refers to the surface of water. That's why it kind of holds its shape very well. And this allows stuff like insects to sort of just like drift on top because of the surface tension. And something that oftentimes happens is they get stuck to the water pretty easily, which can cause a lot of problems over time. Like, I don't know, death or something. But the next property of water is solvent properties. So how is water a good solvent? Well, this has a lot to do with solubility. What can dissolve in water? Well, let's start with salts. For example, sodium chloride, NaCl. Well, NaCl looks something like this. The small one, I'll just like give it some color coding at the very least. I thought drawing these would take a little less time in retrospect, but oh well. Okay, there we go. We're going to say that the tiny green one is sodium, and the big red one is chlorine. And sodium has a positive charge and chlorine has a negative charge. As a crystal, they're very closely associated. But when you add water, we're going to get stuff like this. I'll just draw this out. We're going to get these all dispersed around, and this positively charged sodium is going to have strong interactions with the partial negatives on the oxygen, because negative and positive really like to interact with each other. So the oxygens on water are going to interact with this sodium like this. And in the chlorine, it's going to be the opposite. Since chlorine is negatively charged, we're going to have water interacting with, well, the hydrogens, because these are partial positive. Partial positive. Water likes to stick into crystals and give these sort of shields. And these are called what? Oh God, that's a horrible. There we go. These are called hydration shields. Water likes to surround positive and negative ions in different ways to basically tear them apart from a crystal. And that's how dissolving salt works. And that means it's very soluble. It dissolves very well in water. Now, the next thing we have is polar molecules. So let's do an example. Let's do glucose again. Glucose has lots of OH groups. And what can these do? Well, these can have strong hydrogen bonding interactions with water. Or the oxygen here can interact with the hydrogen on a water molecule. So all of these hydrogen bonding forces and polar interactions can cause this to associate very well in water. So these polar interactions make both polar molecules and salts water soluble. And when we see something that's soluble, another word for it is hydrophilic. And this means water loving, water, philic, loving. So this term you're going to have to know pretty well here, hydrophilic. Some other examples of polar molecules include amino acids, oxygen, and other molecules as well. This oxygen has lone pairs that interact with the hydrogens on water, and amino acids also have their own different interactions, and these make them pretty soluble, or hydrophilic, water-loving. So one thing that's pretty applicable here is transport. So when you have polar molecules or salts, these transport very well in your blood. They're transported freely. When we get to nonpolar molecules, we're going to see how that becomes a little bit of a problem. And that's what we're getting to right now. So an example is fats. And let's just draw a triglyceride here. If we have something like this, this does have oxygen, but it doesn't have those hydrogens. And this means that water doesn't interact as well with these. And this has way more hydrocarbons than oxygens. And these create a very bad environment for water interactions. So when you have something like fats, you have little to no polar interactions. And that means, this means they're not going to be very soluble or insoluble. And another word for this is hydrophobic. So you've heard phobias. That means you're scared of something. So if something's hydrophobic, it fears water, water fearing. Not homophobic, hydrophobic. So water fearing. And this is really relevant when it comes to transport through blood. It's not transported freely in the blood because it's hydrophobic. It's going to create this little layer on top. As an example, I'll draw a glass of water. If you ever poured oil on water, you get a little layer. And this is what would happen in your blood if these fats were transported freely. You'd get this layer. They would all interact with each other, and that's not good. So instead, what you need is fats need to be part of proteins in order to transport through blood. So proteins are soluble. So when you have something insoluble interacting with something soluble, it makes it more soluble. So that allows it to be transported through blood. And an example of this is something like albumin. You need your albumin in order to transport fatty acids. Or something like lipoprotein, like your good and bad cholesterol. This is how your fats are transported throughout your blood. And that is topic 2.3. Okay, now moving on to that was topic 2.2. Moving on to topic 2.3, we're going to do carbohydrates and lipids. And I see we're getting, it's not too bad. We're pretty good on time here. Here are your learning outcomes. So we're going to start with carbohydrates, and here are our examples. These are monosaccharides. We have glucose and fructose, and these are so ubiquitous in everything you eat because of table sugar or sucrose. So starting with monosaccharides, these are soluble, and like I told you before, those OHs on glucose interact pretty closely with water molecules, which is what makes them soluble. They taste sweet, something like glucose, that's pretty sweet tasting. And they act as immediate energy sources for cells. So what does that mean? Glycolysis is an example. Glycolysis is very fast. You can break down glucose real quick to get energy. And this is very important in something like your muscles when you need energy like as soon as possible. So if we look at examples here, we'll just draw glucose a little more specifically here. Now with all of the atoms. Oh, that's not right. There we go. This is the most like non-condensed structure of glucose, and the numbering goes like this. It starts on the right. So carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. So this gives glucose a formula of C6, because there's 6 carbons, H12, you can just count the hydrogens, and O6, 1, 2, 3, 4, 5, 6. And there can be different types of this glucose. But before we get ahead of ourselves, we're just going to look at the numbering here. So carbon 1, this one's pretty important. The position of this OH is variable. And here's how you can tell what the position means. So if it's on the same side as carbon 6, this little methanol group poking out of the ring, if it's on the same side, then that's called beta. Beta glucose is when the OH is on the same side. C1OH, same side as C6. So that's when you get beta. Now, the second position that this OH can have is when it's flipped. So I'll just draw this again. I forgot to draw this little H here. There we go. So now, if we look at C6 and C1, specifically their OH groups, so let's just label these again, they're on opposite sides. And when they're on opposite sides, we call this alpha. And this whole molecule is glucose. So when you see alpha, it means the C1OH is on the opposite side of C6. So when they're on opposite sides, it's called alpha. And it's glucose because of the arrangement of the other OH groups. If any one of these was arranged differently, you would get a different name for the sugar. And let's see. I'm going to show you just an example of what that means. Oh, I guess I don't really need to block this out. We're going to have something like galactose. Since this OH on carbon 1 is variable, I'm just going to draw it like this. When there's a squiggle, it can be either on the top or bottom, so alpha or beta. So far so good. It looks like glucose, except this part. The OH is on carbon 1, 2, 3, 4 is on top instead of on the bottom. Now, if we look at glucose, usually in carbon 4, it's on the bottom. Looking here, it's on the top. And that changes its properties, which is why it's called galactose. Something with a different arrangement would have a different name. That is the point I was trying to make. Also, the arrangement of the carbons in the ring can also give it a different name. Let's draw something like fructose. Well, if we look at this, this looks like it's 5 carbons. Well, it's not. It just has 2 of these methanol groups, which gives it 6 carbons still. This is still C6H12O6, and so is galactose. Sugars can have different molecular formulas, but still be different sugars because of the way their carbons are arranged. So here, it looks like a pentose because it has 5 carbons, pent 5-ose sugar, so pentose. But all of these make it a hexose because it has 6 carbons total. So it's still numbered starting at the first carbon on the right. So it starts with this one, and it still has 6 carbons. So basically what I'm saying is both the arrangement of the OH groups here, as well as the arrangement of the ring, can change the properties and therefore the name of the sugar. Next, we're going to talk about glycosidic linkages. So I briefly mentioned them in 2.1, but here's where we start using them a little more. So let's draw a simple sugar like glucose. Oh, another name of these, by the way, is simple sugar. So if we draw 2 simple sugars here, let's do glucose. This is alpha glucose. I'm going to leave these out for simplicity. When we react these two together, this looks like a condensation reaction. Take this OH, take this other one, or the other way around, and we get something like this. And let's just number this real quick. So starting on the first carbon on the right, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So what's kind of significant about the numbering is we have the carbon 1 OH and the carbon 4 OH combining together. And since it's a condensation, it produces a water. So it's a condensation. And the product is a disaccharide. So this linkage right here is the glycosidic linkage. And it's on carbons 1 and 4. So what would you call this? A 1,4 linkage, or 1,4 glycosidic linkage. Now, there's another term that we need to incorporate into the naming here, and this is alpha or beta. Well, whether it's alpha or beta depends on the position of this OH here. Well, let's look at this. We learned before that if it's opposite to the C6, then that means it's alpha. So that makes the whole thing an alpha 1,4 linkage because of this alpha on the first sugar that's reacting. So this is an example, an alpha 1,4 linkage. Now you can have more than one type of linkage as well. I'll just label this because this is a condensation. Another type of linkage you can have is a beta linkage. So if we were to just draw this again, and instead we have let's see. Well, if we look at the first sugar, first if we number things here, 1, 2, 3, 4, it's still a 1,4 linkage. But if we look at the first sugar that attached with its carbon 1 OH, well, initially it was facing up. And when it faces up, it's on the same side as C6. And when it's on the same side, what did we learn? Well, it's called beta. So this here would be a beta 1,4 linkage. And you see this in things like cellulose. These are more common. You see these in a lot of different polysaccharides. But yeah, this is the basics of a glycosidic linkage and their condensation reaction, so they produce water. Next, we're going to get into disaccharides. So these are soluble. Again, these are still small enough to be soluble, and they act as a transport form. So in plants, you get stuff like sucrose and maltose. And in animals, you get stuff like lactose. And lactose is important in animals because in animals of the female sex, when they're having their offspring, its purpose is to transport carbohydrates into the offspring. So in milk, literally. Let's just draw these out. So let's start with lactose. One sec, I'm just gonna double check something real quick. Right, okay, my apologies. I was drawing it right to begin with. Okay. So this is lactose. And the two sugar units you can see are both hexoses because they have 6 carbons. And the linkage between these, let's see, 1, 2, 3, 4, that's the second one. And it's carbon 1, so it's a 1,4 linkage. And if we look at this here, well, this one originally is opposite to C6, so it's going to be an alpha. So an alpha 1,4, oopsie, alpha 1,4 linkage. And what are the monomers here? Well, the monomers are galactose and glucose. So glucose and galactose make lactose. And you can remember this pretty easily by seeing the lactose in galactose. So combining these together will give you lactose. Let's look at sucrose. This one's a little trickier. So let's draw our fructose. And by the way, you don't need to know how to draw fructose or galactose. You just need to know how to draw glucose and ribose. Just to clarify. So this might look a little weird, and that's because I'm just drawing glucose I'm flipping it a little bit, because the linkage here is actually between carbon 1 of glucose and carbon 2 of fructose. That's why. So the thing here is, oh, 1 second. I'm just going to make sure that I explained fructose properly. Yes, okay. False alarm, besties. So this OH is on the opposite side of the carbon 1. This time it's carbon 2, and the reason why is because it's just the first carbon in the ring. So the first carbon in the ring, the OH is opposite to this one, making it alpha. So alpha fructose combined with, well, this is glucose flipped, and this is C1. If the carbon 1 OH is opposite to this one is also alpha. So this is actually an alpha 1, alpha 2 linkage. This is extra. You don't have to know this one. No worries. All you have to know is sucrose is made of fructose and glucose. That's it. We'll make it a little easier for you guys. Our next one here is going to be maltose. Maltose is really easy. So this is glucose, and this is glucose. Just to clarify one more time, you guys don't need to know the linkages. I was just showing that just to have some kind of application for you guys. It would be good practice. But glucose plus glucose makes maltose. And just quickly going over the linkage here, we start here. So 1, 2, 3, 4, and 1. So it's a 1-4 linkage, and both of these are alpha. Well, the one that matters is alpha because it's C1. C1 is opposite to C6, making it alpha. So it's an alpha 1-4 linkage, but once again, you don't need to know that. As a summary, for disaccharides, sucrose is glucose plus fructose, or fructose. Lactose is glucose plus galactose. And finally, maltose is glucose plus glucose. So one thing you'll find in the disaccharides is that glucose is part of all of them. All of them has at least one glucose. The difference comes in the second one. So galactose makes lactose if you combine it with glucose, and you can remember this with the lactose in the name. Sucrose is when you combine glucose and fructose, and maltose is when you combine glucose and glucose. So that is what you will have to know. Okay, that was super long, but now we're going to get to polysaccharides. And first and foremost, these are poorly soluble. And the reason why is because there's a lot of them, very large chains. When you have a very large chain, those interactions with water don't really matter anymore because it's such a big chain, it clusters together and precipitates, making it very hard to dissolve. Big clumps kind of interact with each other better than water. So, what do polysaccharides do? Well, they act as a structural support or storage form or cell recognition. And we talked about this. This is glycoproteins. So glycoproteins, just draw a quick membrane here, are in our cell membrane, and this is a protein. And you could have something like this. This is a glycoprotein, and these can interact with other cells. And they can create recognition. So this is a lot to do with how your immune system works, a lot to do with how cells kind of know which cells to talk to, I guess. But anywho, one example we're going to get into is cellulose. So what does cellulose look like? Well, it's made of glucose. And together, these create a specific linkage. So if we look at 2, let's number here. 1, 2, 3, 4, and 1. Well, that's a 1,4 linkage. And look, initially this was on the same side as carbon 6, which makes it a beta 1,4 linkage. Now this you have to know. Cellulose is beta, and you have to know this. In images, you might not always see this kind of squiggle form. You might see something like this. Oh, oops. And when you see it this way, it'll look something like this. One really easy way to recognize beta 1,4 linkages is that these alternate. When these alternate, that's when you have a beta. And these are the same. They're just different ways of writing it out. So this is a beta 1,4 linkage, and you should know this. So that's cellulose. Well, what's the function? The function is plant cell walls. And these are not digestible. By humans. something like cows, they can digest it with special bacteria, and they have very complicated gastric tracts because they eat grass, which has cellulose, and they use all of that to create energy. But for us, they are not digestible. Oh, and another thing about cellulose, it's a single chain. This is unbranched. I mentioned this before. And one thing cellulose can do is form a double helix. Which is a very special property it has. Next, we have, oh my gosh, wait, no. My apologies. It does not, that's amylose. Cellulose has no helix. Amylose does. Cellulose looks like that, and it lines up in like basically sheets, and it's what forms the plant cell walls. Sorry, guys. It's so early. Next, we have amylose. So this one's actually super duper easy. So in this, we're going to have the CH2OH group pointing up every time. And that makes it an alpha 1,4 linkage. For amylose, you do have to know this, 1,4 linkage. And just to break it down, if we number this, 1, 2, 3, 4, because this is carbon 1, and this is also carbon 1, it's a 1,4 linkage. And well, let's see. This C1OH is opposite to this, and when it's opposite, it's alpha, making it alpha 1,4. So it's a pretty simple sort of chain, and its simplicity allows it to form helical arrangements. So in plants, it forms a helix. This one is helical, not cellulose. And yeah, so it's a 1,4 glycosidic, it's an alpha 1,4 glycosidic linkage, and its digestibility is actually pretty hard. It's pretty hard to digest. And this actually makes it the main storage in plants, more long term. And its function is energy storage. And it's pretty hard to digest for humans. Whenever I say digest, we're always going to refer to us. But for plants, they're also kind of hard to break down, which makes it a good long-term energy storage. Next, we're going to get into amylopectin. And before I do, this is unbranched. And amylopectin is very similar, but this time it's branched. Crazy. And it looks very similar to amylose. But then, not to throw a curveball, but these can have other kinds of linkages too. So these have two different kinds of glycosidic bonds. We're just going to move this down a little bit. There we go. So let's break this down. Let's look at the first one here. So these are glucose units once again. And the same goes for amylose. So these are all glucose monomers, all of these polysaccharides. And what happens is for amylopectin, if we look at the linkages, okay, well, there's 1, 2, 3, 4, 1. This is alpha 1, 4 because this is opposite to this. So this one is alpha 1, 4. Let's look at this one. Well, 5, 6. There's carbon 6. Okay. This is carbon 1 again. And since it's opposite here, it's also alpha. But instead of 4, it's 6. Well, this makes it an alpha 1, 6 linkage. And this one's actually really important. These ones you have to know, once again. 2 different linkages. It's branched. What's the function? Well, the function in plants is for energy storage too. And this is really important because this is actually easier to digest. And the reason why, for us, is because it's very similar to glycogen. Which is our storage. Very similar to glycogen. So it's easy for us to break it down. And for plants, it functions as a more short-term energy storage. It breaks down a little quicker. But yeah, that's kind of how it works. And when you look at it overall, these are going to be chains like this. And sometimes we're going to have branches. So stuff like this. So one thing I will note is these last two that I went over, amylose and amylopectin, make up starch, like plant starch that we consume, that we use for baking. These are the main components of starch. Now, we have our own starch that we don't get in our diet usually, but it's our own storage form of glucose, and this is called glycogen. And this is also glucose monomers. And this one is branched as well. And it looks the same as amylopectin. So this same exact thing that we saw with the myopectin. This is alpha 1, 4, and this is alpha 1, 6. So once again, memorize this as well. Glycogen has the same types of branching as amylopectin, but the difference between the two is A, its frequency of branching, and B, its function. So starting with its function, in mammals, its function is energy storage. And this is our short-term energy storage. This can be broken down really fast. Glycogen exists in our liver, and when we really need sugar in our blood, then we break down our glycogen to get sugar And the breakdown is through hydrolysis. You use water to break one into two. So then this glucose goes into our blood and we can use it for energy. So this is extremely important in our bodies. And if we draw kind of what it looks like, once again, it's very similar to a myelopectin. And there's one kind of big difference. And it has more frequent branching than a myelopectin. A myelopectin has less frequent branching. So it looks like that. All right, and that is all our polysaccharides. Now we're going to get into lipids. So a lot of this is just an image from the internet. These are the ones we're going to look at. Fatty acids, triglycerides, and phospholipids, and also steroids, just this part. These ones we don't care. Okay, starting with fatty acids, we're going to get into some theory here. So fatty acids are carboxylic acids with very long hydrocarbon chains, about 4 to 24 carbons. What does this mean? Well, what is a carboxylic acid? It is a carbon, double bonded to oxygen, single bonded to OH. And the rest is an R group. So R can be anything. So whenever you have something like this is a carboxylic acid. And this R group is what this is, very long hydrocarbon chains. So lots of C, 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 H, H, H. Tons of that. And that makes it very nonpolar and very hydrophobic, not soluble in water. So there's two main types. Saturated, you need to know how to draw this. The number of carbons on this doesn't matter. As long as it's between like 10 and 20, just a large enough amount such that it looks like one, then you're good. Saturated means there's no double bonds. There's a maximum number of hydrogens. It's found in animals, which means like stuff like butter, steak. Even chicken. It's linear. You can see that it goes in a straight line, so it's linear. And it's solid at room temp. Why is it solid at room temp? Well, the reason why is because it's linear. So here's an analogy. Think of a box of spaghetti noodles. Spaghetti noodles are super straight, so they pack really well together. And the same can be said with this. This is basically a fatty acid. And when we have a lot of these also pack together because they're linear. And that means they have lots of interactions between them, intermolecular forces. And these would be London dispersion forces or van der Waals. And when you maximize these van der Waals, you get very, very stable interactions, which means it takes more energy to break them apart. So just to illustrate this, we have strong IMFs takes a lot of energy to separate them. So because of this, it's solid at room temperature, which is why we have stuff like butter. Moving on, we have the second type, unsaturated, and that means it has one or more double bonds. And there's two different subdivisions of this. There's cis, unsaturated, and I'll explain this in a second. Here's a double bond. And when it's cis, these carbons in the double bond are on the same side. So cis means the carbons are on the same side. And when you get stuff like this, it creates a bent conformation. So in reality, it creates something like that. We call it a kink, which is why phospholipids are pretty relevant here in terms of membrane fluidity. But basically, when there's a double bond, it bends. And this makes it liquid at room temperature. And this is why, because it's bent. Remember what we said about the spaghetti box here? Well, now these are bent. Think of these as silly straws. And then when we have a lot of silly straws, we don't have very many interactions. So we have less intermolecular forces, which means it takes less heat to separate. So these are going to be liquid at room temperature. Now these are found in plants. This is the fat storage in plants, and this is found in oils. Oils are liquid. So they're found in stuff like plants. Next, we're going to talk about trans unsaturated. And this is the opposite of same side. This is when, let's just draw this out here. When we have a trans double bond. Trans looks like this. And it's when these here are on opposite sides. When they're on opposite sides, it gives you trans configuration. So when we have trans unsaturated, they're largely linear. These remain almost basically as linear as saturated fats. And what does that mean? Well, because it's linear, it's solid at room temperature. And we talked about this before. They pack very well, which means there's a lot of forces, which takes more heat to break or separate. Now, these are different from other ones because they're not found in nature. They're synthetic. And examples of this is margarine. Stuff like, I can't believe it's not butter. Well, it's not. And the reason why is they take oils and they add a bunch of hydrogen and they take some of the double bonds and make them trans, from cis to trans, which makes them solid. It's basically a way to make butter vegan by not taking it from animals. And these are really bad. Both saturated fats and trans fats are bad for you. And we're going to get into why. I'll clarify as well, you don't need to know how to draw these. You do need to know how to draw this one, though. So yeah, that is our basic fatty acids. Next, we're going to move on to when these fatty acids combine. So we mentioned these before. These are triglycerides, the largest class of lipids. And the purpose for this is long-term storage. Long-term. This is really important. Polysaccharides are short-term, but triglycerides are long-term. In animals, they're stored as fat, a solid, in adipose tissue. In plants, they're stored as an oil because unsaturated fats come from plants. And they don't pack very well, so it takes less energy to separate them. So yeah, triglycerides themselves are formed through condensation. And this condensation creates something called an ester linkage. What is an ester? Well, R1, C, we have our carboxylic acid group. Let's add an alcohol. Let's add one. Well, what's going to happen? This OH and this H in this specific arrangement are going to leave as water, and the result is going to be R1, C, double bond O, O, R2, plus water. So it's a condensation. It produces water. These leave as water, the ones that are boxed in red. And then the resulting structure, it looks like a carboxylic acid, the double bond to oxygen and the single bond to oxygen, but instead of H, it's a different hydrocarbon. So this is what's called an ester linkage, this right here. So like I said, acid plus alcohol gives you ester. Triglycerides are made up of 3 fatty acids and glycerol. And we're going to get into how this is made. First, I'll show you what glycerol looks like. This is glycerol. And in biology, it's drawn like this. Same thing. Same exact thing. These 3 lines mean they're equivalent. And for all my chemistry besties out there, the name for this is, well, it's a propane with 3 OH, making it 1,2,3 propane triol. So it's a pretty specific structure, but for all my bio kids that hate chemistry, I relate. It's just 3 carbons with an OH on each carbon. Okay, now we're going to get into the synthesis of a triglyceride. And this is a pretty, I think you might have to be able to recognize this on an exam. So I'll draw this in terms of carbons. So this is glycerol right here. And this is going to react with three fatty acids. Oops. There we go. These are all going to produce condensation reactions. And what they're going to do is the result is going to look like this. This is a triglyceride. Now, if you're wondering, these R groups can be the same or different. It doesn't really matter. And this creates a lot of variability, which is why it's the largest class of lipids. It can be the same or different. So this is going to be a triglyceride. And don't forget, it produces another thing. Well, what happens when a condensation reaction occurs? You're going to get water. And there are 3 pairs of H and OH that are removed. So that's going to be 3 water, because there's 3 kind of condensations happening at the same time. So another word for this is esterification. And this just means it forms an ester. Esterification, form ester, whatever. You need to know the condensation part, though, that this is a condensation reaction. Okay, now that we know how triglycerides basically work, we're going to talk about their relevance in health and disease. And this is all going to come through the perspective of cholesterol. What is cholesterol? Well, cholesterol is a steroid, and it looks like this. You don't need to know how to draw it. Don't even worry. This right here is cholesterol. Now, cholesterol on its own, I just want to say, is not bad. In fact, it's really good. It has so many different functions. It's involved in membranes. It kind of sticks into membranes to help with fluidity. It helps make hormones. Hormones like cortisol or estrogen. These are super important. And you also have bile. Bile is what helps your fats be absorbed and digested. And this is all because of cholesterol. They form bile salts that help emulsify your fat. Now, cholesterol, in order to transport it throughout your body, it requires a protein. Do you know why? The reason why is because if you look at this OH group, it's the only polar part of this entire molecule. The rest of it is very kind of non-polar because it doesn't have any electronegative groups or any sort of polarity to it. Because of that, this makes it insoluble or hydrophobic. So you need to attach it to a soluble protein in order to transport it throughout blood. So that's why it requires protein. And the type of protein cholesterol is transported in is called lipoprotein. And this is where we get our good and bad cholesterol. This is what people are talking about. When they say good cholesterol or bad cholesterol, they're talking about the protein. So the first one we're going to talk about is good cholesterol, HDL. And HDL stands for high density lipoprotein. I'm just trying to find a place to write this. I'll just do this here. High density lipoprotein, and the L stands for lipoprotein. And then good cholesterol, the purpose of high density lipoprotein is taking cholesterol from your tissues, your muscles, your adrenal glands, any tissue that isn't your liver, and it takes it to the liver. So the purpose of it is to offload any extra fat in your cells and sends them back to the liver. So this is really good because it helps preserve your cell's ability to kind of keep going. You know what I mean? Next, we have low density lipoprotein, and this is bad cholesterol. And the reason this is bad is something we'll get to soon, and it sends cholesterol from your liver to your tissue. So it takes cholesterol produced by your liver and is like, okay, I'm going to give this to all the cells that need it or don't. It doesn't really care. Now, the type of fat you consume in your diet will affect the cholesterol lipoproteins. So if you have cis-unsaturated fats, well, what are these again? Well, these are the ones with the bends because of that double bond. They're the ones with the bends. And these are super good. These are really good because they lower your low density lipoprotein and raise your high density lipoprotein, which means you have less cholesterol leaving your liver to offload to your other cells, and you have more of it being sent back to your liver. And this is good because then all your cells are kind of working in harmony. Then we have trans-unsaturated and saturated. And these are going to raise LDL and lower HDL. And this is bad. What are these again? Well, trans-unsaturated looks like this. And saturated just looks like this. And these are both linear, and they're solid at room temp. And these are bad because they increase cholesterol going to your tissues instead of back to your liver. And this isn't good because maybe your cells don't want any more cholesterol, but they're getting in them anyway. And that creates a lot of problems. One of them is called coronary artery disease, or another term you've probably heard of is coronary heart disease. and that's right here. So this is what people talk about when they say eating trans fats or saturated fats will increase your chance of heart disease. This is what they mean. This is caused by a very long word called atherosclerosis, or plaques. And these are blood clots, and they are a result of high LDL. So I'm going to talk about this a little bit more. Let's say you have an artery. A nice little artery here. Looks clean, looks nice. Well, let's say you've been eating a lot of saturated fats and trans fats. And when you do that, there's a lot of LDL floating around. The reason why is at some point when you have enough LDL, your cells say stop, no more. So it no longer accepts the LDL, so it builds up over time when you consume a lot of fats, saturated fats, or trans fats. And when this happens, this creates a problem because let's say you cut yourself. Well, when you cut yourself, you hurt a blood vessel. You hurt this blood vessel. It creates a little cut. So when you get a cut, your immune system responds because your body wants to fix it. So these are when the good guys come in. Well, when the good guys come in, they're out here fixing this little cut. And these are going to be white blood cells or macrophages. Here's where the actual problem comes in. We have all this LDL, right? And it's floating around and it wants to go somewhere. White blood cells, they're going to take the LDL. They're going to take it because LDL now has a bunch of cells that it says, oh, they want some. They don't have any. So I'm going to offload myself into them. So what does it do? It's going to get into those white blood cells. They're going to fill up with this LDL, with this bad cholesterol. And what's the result? Well, you're going to get a bunch of these cells all bloated up, and they're going to be filled with cholesterol. And this is going to form a plaque. And what happens over time is your blood vessel regenerates itself and it forms on top of this. So this can't be removed, or it's really hard to remove. So now you're going to get constriction. And what's this going to lead to? Blood clots. It increases your chance of getting blood clots. And what does this cause? It can cause stroke, a blood clot in your brain, and heart attack. A blood clot in your vessels that plugs blood from coming to your heart. So this is why this LDL is so bad, because these blood clots are so much easier to form now over time that they can increase your likelihood of these things so easily. And this is heart disease. This is what happens with coronary artery disease. We're almost done, guys. Don't worry. Okay, finally, we're going to compare sugars and fats. A quick little comparison. And there are 5 kind of axes to compare them with, and the mnemonic you can use is sodas. Sugars and fats, they like sodas or something like that. But the S stands for storage. And here's how you're going to compare them. Well, for sugar, it's short term. I've talked about this before. When you need energy really quickly, that glycogen in your liver is broken down into glucose ASAP. Super easy. Fats, well, this is more long term. Only when you run out of your short term storage do you start using your fats. Think of sugars as like a wallet. You're going to have some money in your wallet when you go to the store, and it's for quick and easy access. You need your card to pay for something, get it from your wallet. Well, fats, think of them as a safe. If you just won the lottery, you're not going to store your millions in your wallet, right? That's not really practical. So you're going to use a safe for storing these things. And whenever you really, really need them, in the case, maybe say you don't have money in your wallet anymore, you're going to go to your safe and get all the money from there. So you're going to go to your fats and get energy from there when you can't get your sugars. Hopefully that made a little bit of sense. That's a pretty good analogy. Next is osmolarity. And this just means, does it affect your cells in terms of their hypotonicness or isotonicness? I think you've talked about this with plants in your bio course. So with sugars, yes, it can affect osmolarity. And this basically means sugar is able to influence osmosis. If your cell has a lot of sugar, but there's not very much sugar in the blood, your cell is going to fill with water to balance the concentration. And that's going to create a bloated cell. So that's what I mean by osmolarity. And the reason why is because sugar is soluble. Monosaccharides, disaccharides, these are soluble things. And when they're soluble, they can change the concentration of solute in your blood. Well, when you have fats, these aren't very soluble. So it won't change your osmolarity. Fats like to clump up into big droplets. And when they do that, they don't really change concentration of things in your cell. They just kind of stay in one little place. And because of that, they don't change concentrations enough for water to be like, hey, I'm going to leave or head in. You know what I mean? So fats don't change osmolarity. Next, the D, digestion. Sugars are very easy to digest. And the reason why is because they're transported very easily through your blood, and the reactions to break them down is very simple, which is what makes them short term in terms of storage. Fats, these are harder to digest. That looks like an F. There we go. The reason why is because, well, they're nonpolar. They're going to clump up in your stomach and intestines. So you're going to want something to unclump them, and that requires bile. And that makes it more complicated. And the reactions required to break down fats are also more complicated. So it's overall harder to digest. It takes more energy, which is why it's a long-term storage. Next is ATP gross. Basically, what that means is, how much ATP do you get? Well, for sugars, let's pick an example. If you have glucose, you're going to get 32 ATP. If you have a fatty acid, depending on the length, sometimes you can get over 100 ATP if it's big enough. And the reason why is because fatty acids have a lot of those carbon-hydrogen bonds, which are very reduced. Those are very easy to take electrons and energy out of. And glucose doesn't really have much of that. It has a lot of carbon-oxygen bonds, which are not reduced. And because of that, it doesn't give as much energy as you would have liked. So fats give way more energy than sugars. They're very efficient in terms of fat, which is good for long term. Solubility. Well, sugars are very soluble because of all those OH groups and the ability to form hydrogen bonds with water. And fats are insoluble because they have so much of that hydrocarbon that this part doesn't matter. This part is what matters. So it creates these interactions that are not compatible with water, and as a result, make it insoluble. They clump up in liquid. So yeah, that was your comparison. Remember the mnemonic sodas, and that'll be an easy kind of way to compare things. Okay, last kind of main thing. Yeah, this is the last main thing. Body mass index. So this is a very kind of controversial way of kind of determining the health of an individual based on their weight. But you do have to know this for the course. So the formula, BMI equals your weight, and the unit here is kilograms. And you divide that by your height in meters. And that's going to be squared. So that's the formula, weight over height squared, both in metric units. And a way you can read this is with something called a nomogram. I think that's what it's called. These values come based on height and weight, as I said, and based on their results, you can get different sort of thresholds. And these colors will tell you what class it falls into. So if it's red, then let's look at, if this is red, for example, let's look at the red one here. Well, class 3 obesity. Okay. And the border here is determined by specific values. So these are basically arbitrary borders that if you look at them, you'll see kind of a linear relationship. If you get someone's BMI in a question and you need to determine what kind of, I guess, like where they are in terms of their health, if they're anywhere kind of above 40, because all of these are BMI values, if they're anywhere above 40, then that's probably class 3 obesity. Anywhere between 35 and 39, so anywhere between 35 and 40, well, that's going to be class 2, and you just have to look at the values on here. And through that, you'll understand where they are. Now, just talking about the ramifications of this, this is not accurate for pregnant people or athletes. And the reason why is because they have a different fat composition. And also muscle plays a part. There are things affecting their weight that isn't their fat. And honestly, I'm just going to say this is not accurate, period. People have so many different body types, different body shapes, and people can have such variant metabolisms that it's almost not important to talk about BMI. But doctors are still doing it anyway. But I think it's not very useful because it basically puts a label on something you can't necessarily control. So just for the future, if you guys ever hear people talking about BMI, just know that it's not very useful in telling you how healthy you are or where you are in terms of, I guess, I don't know, your health. Same thing I said before, but whatever. Okay, last two topics. These are going to be pretty quick, I hope. So 2.4 is proteins. Here are your learning outcomes. You can look at those later. Starting with the monomer, so amino acids, we talked about these already. They have that central carbon, the amino group, the COOH group, and the variable R group. Let's draw one real quick. We got that COOH. What else do we have? We have an H2N, which is the same as NH2, it's just reversed because it's directional. And what are we going to have? We're going to have an R group. This can be anything. And then we're going to have an H. That is your generic amino acid. Know how to draw this. New information. There are 20 universal amino acids present literally in every living thing pretty much. And they are separated into different categories. So they can be polar. And in the polar category, we can have charged or uncharged. And then we can have non-polar. So there are lots of different classes, lots of different kinds of amino acids you can have, and thus different interactions. So properties of amino acids are determined by their R group. So like I said, these classifications, polar, charged, uncharged, nonpolar, they determine the properties of that amino acid, especially in a protein, because proteins fold based on the composition of their amino acids. So depending on which R groups they have, that's going to affect how they fold. Okay, peptide bonds. We've seen these before. I briefly did a reaction. Let's do another one. So we're going to draw two amino acids here. And we're going to separate these by R1. I think it'll be a little bit easier if we just draw it this way. And this is R2. So what's going to happen? Well, oh, this is a lot better. Yeah, that's the carboxylic acid group. We remember that one from fatty acids, where it's the C double bond O single bond OH. Well, what's going to happen when these condense? Because this is a condensation reaction that we're going to talk about here. This OH and this H are going to leave, and they're going to become water. So this is just the water leaving. And here's what we're going to have now. This is going to be a dipeptide. Anytime, and I mean anytime, you see this kind of bond, C double bond O, NH, that is called a peptide bond. Be able to recognize these on exams, on questions. Practice the absolute heck out of these. These are going to be really important. Peptide bonds. So what is this called? This is called condensation, because it produces a water and you're forming something bigger from something smaller. Well, sometimes you're going to want to break proteins down, say when you're digesting them in your stomach. And a way to do this is through what's called hydrolysis, which requires a molecule. The reason why is this water gets in here and attacks, which kind of breaks them in half. It's like basically a pair of scissors, and it gives them that OH group and that H group. That's what the water does in hydrolysis. So be able to draw a reaction out. So I'll just highlight this for you guys. Know how to draw this, because this is really important both in genetics, in digestion, so many different things. And these are how peptide bonds work. Another thing I will say is that polypeptides or proteins run N to C. What does that mean? Well, in this dipeptide, we have this free NH group, and this is called the N-terminus, because it's terminal at the end and it's an amino, or amino terminus. What's this thing? Well, this is the C-terminus, or carboxy terminus, because it's terminal at another end and it's a carboxyl group, so C-terminus. And these are important because anytime proteins are made, they're going to be made in the direction of N-terminus to C-terminus. It's going to start with the N group, and then it's going to keep going that way, N to C. So this will be really relevant in transcription, translation, all that fun stuff. Next, we're going to get into protein structure. We're going to absolutely hammer through this. Primary, well, this one's easy. This is just amino acid chain. What amino acids are in a polypeptide. Poly meaning more than 2 or 4. Which ones are in there? The R groups are variable, and yeah, it's basically just the order of amino acids. Secondary structure, or 2 degree symbol, there's 2 different kinds. The first one is called an alpha helix, and the second type is called a beta sheet, or a beta pleated sheet. You could see either beta sheet or beta pleated sheet. They refer to the same thing. And these are dependent on hydrogen bonding patterns. Not between the R groups. This is going to be just between the NH, the CO, the central carbon, not the R group. It's going to be specifically between the backbone. So anytime you see alpha helices or beta sheets, they're going to be formed from backbone hydrogen bonding interactions. So in helices, they form in a specific order where it's one binds to the next every 3 amino acids, and it forms this kind of twirl, I guess. And it's a right-handed helix. And they look like this. And then for sheets, it's going to be very kind of linear. Every one amino acid has another to bind with, or hydrogen bond with. And these interactions create a sheet-like pattern, or beta sheet. And it looks like that. These are really important because they are the foundation of our next structure, which is tertiary structure. So this is the fully folded protein, and the type of folding in tertiary structure depends on R group interactions. So these can take different forms. They could be ionic, where a positive charge interacts with a negative charge. These could be polar, where a delta positive interacts with a delta negative, or partial positive and partial negative. Or they can be nonpolar interactions, where they have a lot of van der Waals happening between them. Very strong associations. So these R group interactions will tell you how the tertiary structure is going to end up being. So it's this full kind of complement of everything. This is your tertiary structure. Kind of a bad drawing, but whatever. So all proteins have primary, secondary, tertiary, all of them. Now, this is the one that not all proteins have, and this is quaternary structure, or 4 degree symbol. This is whenever proteins or tertiary structures have multiple subunits to them. Proteins have multiple different monomers, or when a protein has prosthetic groups. So a really good example of this is hemoglobin. I'm just going to do this in different colors. They're all kind of the same, and one really special thing they have is heme, and this is what allows oxygen binding. This is what allows hemoglobin to transport oxygen. And then this has 4 globin subunits. So here's the thing. If a protein has multiple subunits but no prosthetic groups, it has a quaternary structure. Or when it has just prosthetic groups, nothing else, maybe one subunit, also quaternary, or both. So you can have either one of these. So it can have more than one polypeptide bound as an overall protein. So they don't have to be covalent bonds. They can be just very strong interactions. So you can either have multiple subunits, or you can have prosthetic groups, or both. I feel like this is kind of ambiguous. We're just going to highlight this. Not all proteins have this. So remember this. Proteins can just have a tertiary structure and be completely functional. Now we're going to talk about factors affecting function. So this is denaturation. So what is denaturation? A structural change in a protein resulting in the loss of its function or functions. And there are two factors involved, temperature and pH. How does temperature work? Well, in a protein, we're going to have specific interactions that help keep it together. When you add energy or heat, these interactions can break. And the result will be an unfolded protein because those interactions are broken. So this causes unfolding or partial unfolding. And because of this, there's a loss of function. It doesn't perform its function anymore. Next, we have pH. This one is a little more specific. So proteins and enzymes have active pHs. And this just means, let's say this is a protein. Some of them may have protons or not. Some of them can be acidic or basic, and the pH affects how protonated they are, how many protons they have. And this affects its structure because some of it allows it to have specific interactions. Let's say we change the pH. Make it more acidic, for example. Well, all those protons are going to leave and create a negative charge, which is going to change interactions. Because what they did was release their protons. And it's going to cause unfolding because some interactions were broken for this and this. So pH can basically change how much hydrogen is on a protein, and it can be different. So if there's hydrogens that aren't supposed to be there, or hydrogens that are not there when they're supposed to be, then that causes change in function or loss of function or denaturation. Next, we're going to talk about the central dogma of gene expression. So we talked about this already. We have DNA. Undergoes transcription to become mRNA or messenger RNA, which then undergoes translation to become a polypeptide, and then after folding, a protein, a functional protein. So this is the central dogma. This is generally applicable to most things. One gene will give you one polypeptide, but there are some exceptions, and you have to know these. So gene splicing. Sometimes genes can be moved around. And when they're moved around, they cause different kinds of expression. So if you move around a gene, maybe some amino acids may be in different orders, and that creates variation in your protein. Functional RNA synthesis. Sometimes the RNAs you make aren't going to be used for translation. Some of them are going to be there because you need them for other purposes. An example is ribosomes. Ribosomes have RNA in them that are functional. tRNA. This is what allows you to make proteins through those tRNAs and amino acids. And lastly, you have genetic mutation. And genetic mutation is when you change nucleotides in some way, either changing a base, rearranging them, whatever way, that causes variation in how proteins are synthesized. So these factors, mutation, functional RNAs, gene splicing, are what can act as exceptions to the central dogma. Okay, next we're going to talk about the proteome, and this is really quick. So proteome is all the proteins expressed in an organism. So it's the totality of your proteins that are possible to make from your genes. And they are unique to the individual because individuals can have different mutations, different kinds of variations in their genes, in their genome, which is pretty obvious. We're all kind of unique genetically, and that creates variation in your proteome. So what does this mean? Why is a proteome more complex than your genome? Well, because of those three factors acting as exceptions that we just talked about. So it's actually in this image. Splicing. There's mRNA editing as well. There's also post-translational modifications. So anything you do to the protein, you could add sugar to make it a glycoprotein. And many other examples. Splicing is one of the three factors. There's also functional mRNA synthesis. Well, I guess that's not very important in variation. It's more so when RNA isn't required for protein. And then you also have mutations. So changes in this will cause complexity. So yeah. Next, we're going to briefly go through protein function. So there's a fun little mnemonic that kind of might not be the most PG, but whatever. It's shits me. And this is on BioNinja, so don't blame me for this. If you want to remember all the axes of function, you can just remember this mnemonic. So the first one is structure. And collagen is used to structurally support different tissues in your body, such as lungs, or even your bones. That doesn't make sense. Your skin. That's a little better. Or, just a fun fact, spider silk. That's made of proteins that are involved in structure. Next is hormones. There are some hormones in your body that are made of polypeptides, and the main ones are insulin and glucagon, and there's a bunch more. But insulin and glucagon are made of protein. So they're pretty important in your body's homeostasis. Now you have immunity. So immunoglobulins or antibodies. You've heard of antibodies, probably. These are very important in your immunity, and they're made of protein. So they're required for cell recognition, immune response, so many things. Next is transport. So transport proteins, we talked about hemoglobin, transporting oxygen. And I mentioned albumin, and this is for transporting fatty acids. They also have a sensory function. Rhodopsin is in your eyes, and it helps you see color and a bunch of other things. Next, we have movement. So actin and myosin, these are part of your cytoskeleton. They help your cell move and also move things within your cell for ease of access. Then we have enzymes. So Rubisco, you probably heard about this one, photosynthesis. This is in your Calvin cycle. Well, I guess not yours, in a plant's Calvin cycle, because we can't photosynthesize. And then we have catalase, and this is used to do a lot of things. And an example is break down H2O2. So when you put hydrogen peroxide on a cut or something, you have catalase in your skin to break it down so it doesn't hurt your cells. So yeah, these are all of the different kinds of functions that you have to know that proteins do, and the mnemonic is shits me. They could have picked a better order, I think, but whatever. Okay, last topic. This is very short. Enzymes. Okay, so you know about enzymes. You've learned a lot about them. What is an enzyme? Well, all enzymes are protein, and they're biological catalysts that speed up a reaction. And how do they do this? They lower the activation energy for a reaction to happen. And another thing about them is that they can be reused. So they don't get used up in a reaction. For names, they're usually named based on the substrate, and the substrate means reactant, so whatever is reacting, plus the term ase. So lipase is an example, and what does it break down? What do you think it breaks down? Lip sounds like lipids. And what lipase actually does is it breaks triglycerides into fatty acids and glycerol. So yeah, the naming is pretty intuitive sometimes. Now we're going to talk about enzyme action. So, catalysis. This just means the reaction, and it's usually in solution. So, in water. Very rarely or at all do you see enzymes not doing things only in water. So, enzymes can be free or membrane bound, so they can be floating around in solution. Oh my god, I keep doing the little loop. Or they can be membrane bound. And the catalytic rate of it, so the reaction rate, my abbreviation for reaction is RXN, determined by collisions. So I'm going to talk about collision theory in a second. But before I do, I'm just going to write out the general sort of reaction that an enzyme has with the substrate, just to jog your memory a little bit. So we have an enzyme. Let's make a really simple one. We're going to call it E. And this is the active site. The active site is highly specific to a substrate. So we have a substrate, or reactant, S. What does it do? Well, it forms an enzyme substrate complex. What's next? Well, catalysis occurs, the actual reaction. Let's say it splits it in half. We're going to call it the product. So this is the enzyme product complex. And then the products leave. And as you can see, the enzyme doesn't change at all. It's never used up. And something we're going to talk about in terms of how things react is collisions. So in a solution, you're going to have things just kind of moving around and bumping into each other. And some things are going to bump in just right to cause a reaction. And there's a specific probability associated with collisions. So the extent that collisions create a good sort of reaction is collision frequency. How do we increase collision frequency? Well, concentrations. If you have more of something, it'll have a bigger probability of colliding with an enzyme. Temperature. Temperature can speed up the energy between different particles, and they can increase the amount of collisions they have, which increases the probability of a certain collision happening. So enzymes operate because of all these collisions, and they can operate better if there's a bigger concentration, or maybe even if you have more energy. It depends. Okay, next is substrate specificity. So we mentioned the active site and how the active site is very specific to its substrate. The active sites bind substrates with high specificity, and the actual structure of the active site depends on the tertiary structure of the enzyme. And that makes sense because that depends on the different R groups in the amino acid chain and the different secondary structures. Denaturation will change the enzyme structure and change the active site, which means no more binding. So let's just draw this here. Enzymes are like any normal protein. And when you change pH or temperature, or both, you can cause a no longer active site. So enzymes can be affected just like proteins, and their function is also affected. So substrates can no longer bind. And here's where we get to enzyme activity. So this is affected by temperature and pH, same as protein. pH, like I said, changes the hydrogens on different groups, which changes interactions, and temperature can increase the energy so much that different intermolecular forces break, and that causes a lot of problems with the interactions, which will cause denaturation. Another one that's new, brand new, is substrate concentration. So I was talking about this in terms of collisions, and we're going to see this in action with a little graph. So this means concentration of substrate, and this is activity. So we're going to see that when we increase concentration, activity goes up like this because the collisions increase, which means you can get more effective collisions. And this just means reaction. So yeah, enzyme activity is affected by the ones that affect protein and also concentration because of collisions. Okay, this is what you've all been waiting for. We are finally done the session. It was a lot of content, but we got through the bulk of it, and this is a very content-heavy topic, so I wanted to thank everyone for participating, and I will end the recording now.